In the heart of post-war London, a scientific revolution was quietly brewing. The year 1951 marked a pivotal moment in the quest to understand life itself. At King's College, the air was thick with ambition and the scent of old books and chemicals. A brilliant chemist and X-ray crystallographer named Rosalind Franklin arrived to lead a new research project. Her task was to study the structure of a mysterious molecule, deoxyribonucleic acid, or DNA. Franklin was a master of her craft, having honed her skills in Paris. She brought an unparalleled level of precision and expertise to the London laboratory. The laboratory at King's College was equipped with state-of-the-art X-ray diffraction equipment. Franklin immediately set about refining the techniques for studying DNA. She designed a way to control the humidity of her DNA samples with remarkable accuracy. This led to her first major discovery. DNA existed in two distinct forms. She labeled them the crystalline form and the wetter B form. This distinction was crucial, as previous researchers had been working with a confusing mix of both. Franklin decided to focus her attention on the B form, believing it held the key. Working alongside her was a doctoral student, Raymond Gosling. Together they meticulously prepared tiny, pure fibers of DNA. These fibers were mounted in a tiny brass holder, called a collimator. The collimator was then placed inside a specialized X-ray camera. A fine beam of X-rays, narrower than a human hair, was aimed at the DNA fiber. As the X-rays passed through the fiber, they scattered in a predictable pattern. This pattern was captured on a piece of photographic film placed behind the sample. The process required immense patience and stability. The exposure times were incredibly long, sometimes lasting over 100 hours. Any vibration could ruin the delicate diffraction pattern. In May of 1952, after one such marathon exposure, a truly remarkable image emerged. It was the 51st photograph of its kind taken in their series. They simply labeled it Photo 51. The image was breathtakingly clear. It showed a distinct X shape, a pattern that screamed Helix to a trained crystallographer. The angle of the X revealed the steepness of the helical coil. The horizontal bars, or layer lines, that formed the X were like rungs on a ladder. The distance between these layer lines indicated the distance between the repeating units of the molecule. Franklin could calculate the precise dimensions of the DNA molecule from these patterns. She determined there were likely 10 units per complete turn of the helix. Crucially, the fourth layer line was conspicuously missing. This absence told Franklin that the structure was a double helix, not a single one. It was a subtle clue, but one of profound significance. He also deduced that the phosphate backbones must be on the outside of the helix. This meant that genetic information, the bases, were protected on the inside. Franklin meticulously recorded her findings in her notebooks and internal reports. She was cautious, a true scientist, wanting to gather more evidence before publishing. Meanwhile, at Cambridge University, two other scientists were also racing to solve the puzzle. They were the ambitious James Watson and the brilliant Francis Crick. They were model builders, using theoretical calculations and educated guesses. But their models had failed because they were working with incorrect data. They lacked the hard, experimental evidence that Franklin was producing. The head of Franklin's lab, Maurice Wilkins, had a strained professional relationship with her. Wilkins was also working on DNA, but his images were not as clear as Franklin's. In early 1953, James Watson visited King's College. Without Franklin's knowledge or consent, Wilkins showed Watson photo 51. For Watson, saying the image was a moment of instant revelation. The unmistakable X pattern immediately confirmed the helical structure to him. He later wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race." Watson sketched the pattern on a newspaper to remember it. He rushed back to Cambridge, his mind buzzing with the new information. The image provided the crucial framework they had been missing. Shortly after, Crick gained access to one of Franklin's detailed internal reports. This report contained her precise measurements and calculations about the DNA molecule. It included the dimensions of the unit cell and the symmetry of the molecule. This data, combined with photo 51, was the final piece of the puzzle. Watson and Crick now had everything they needed. They furiously began building a new model, this time with confidence. The structure they proposed was a right-handed double helix. It elegantly explained how DNA could store and replicate genetic information. The two strands were complementary, like a photographic positive and negative. In April 1953, the journal Nature published 3 back-to-back articles on DNA. The first was Watson and Crick's famous paper proposing the double helix model. The second was a paper by Wilkins and his colleagues. The third and final paper was by Rosalind Franklin and Raymond Gosling. Franklin's paper, with photo 51 included, provided the essential experimental support for the model. However, its placement made it seem like a mere confirmation of Watson and Crick's theory. Watson and Crick's paper included a subtle, understated acknowledgement. They mentioned being stimulated by the general knowledge of Franklin's unpublished work. The true extent of their reliance on her data was not made clear. Franklin, a person of immense integrity, did not protest. He moved on from the toxic environment at King's College. She began a new, more productive chapter of her career at Birkbeck College. There, she led a team studying the structure of viruses. Her work on the tobacco mosaic virus and poliovirus was groundbreaking. She produced stunningly detailed images of viral structures. Tragically, her life and career were cut short. Rosalind Franklin died of ovarian cancer in 1958 at the age of just 37. Her illness was likely exacerbated by her extensive work with X-ray radiation. 4 years later, in 1962, the Nobel Prize was awarded for the discovery of the DNA structure. The prize went to James Watson, Francis Crick, and Maurice Wilkins. Rosalind Franklin's name was barely mentioned. The Nobel Committee does not award prizes posthumously. For many years, her critical contribution remained a footnote in history. The full story began to emerge with the publication of Watson's book, The Double Helix, in 1968. His account, while popular, portrayed Franklin in a negative and inaccurate light. This prompted others who knew her, like Francis Crick and Aaron Klug, to defend her legacy. Historians and scientists began to re-examine the events of the early 1950s. Franklin's own notebooks revealed the depth and clarity of her understanding. It became clear she was not just a technician, but a leading intellectual in the discovery. She was on the verge of solving the structure entirely on her own. Today, Rosalind Franklin is celebrated for her pivotal role. Her story has become a powerful example of the struggles faced by women in science. Photo 51 is no longer just a piece of data. It is an icon. It represents a crucial moment when humanity first glimpsed the secret of life. The sharp black cross on the photographic plate was a Rosetta Stone for our own biology. Franklin's meticulous work laid the foundation for the field of molecular biology. Her legacy is not one of victimhood, but of scientific excellence and integrity. The double helix is perhaps the most famous molecule in the world. Its discovery transformed medicine, genetics, and our understanding of evolution. And at the very heart of that discovery is a single, ghostly photograph, Daiso. Photograph taken with skill, patience, and profound scientific insight. A photograph that truly unlocked the code of life. The initial setup in the King's College lab was challenging. Franklin had to build and refine her own instruments. She used a fine-focus Raymax X-ray tube. The DNA fibers had to be perfectly aligned. Even the slightest misalignment would blur the resulting image. The lab was housed in the basement of the college. It was a damp and somewhat gloomy environment for such precise work. Franklin's dedication, however, was unwavering. She often worked long hours, perfecting her experiments. Her notebooks were filled with meticulous observations. Each diffraction pattern was a small piece of a larger puzzle. Photo 51 was the piece where everything clicked into place. The clarity of the B-form pattern was unprecedented. Wilkins had been unable to achieve such a clear separation of the A and B forms. Franklin's careful control of hydration was her secret weapon. This allowed her to isolate and photograph the B form in its pure state. Diamond shapes formed by the reflections were also significant. They indicated that the backbone of the helix was on the outside. This was a counterintuitive idea at the time. Most scientists, including Watson and Crick initially, assumed the opposite. Franklin's data corrected this fundamental error. The day Wilkins showed Watson the photo was a breach of scientific etiquette. Unpublished data is considered private property until publication. Watson immediately understood the implications of the helical pattern. Crick, with his background in physics, could interpret the finer details of the diffraction. Together, their skills were perfectly complementary. They realized the two strands must run in opposite directions. This anti-parallel structure was another key insight. Their final model was elegant, simple, and profoundly powerful. It was built from metal plates and clamps in their university department. The model stood nearly 6 feet tall. When Franklin finally saw the model, she immediately accepted its correctness. She could see how it fit perfectly with her own experimental data. There was no bitterness, only the satisfaction of a scientist seeing a problem solved. The move to Birkbeck allowed her to be the leader of her own group. Her research on virus structures was equally pioneering. She collaborated with a team that included the future Nobel laureate Aaron Klug. Her work laid the groundwork for the field of structural virology. The discovery of the double helix was a turning point in the 20th century. It opened the door to genetic engineering and modern medicine. The story behind the discovery is a complex human drama. It speaks to the nature of competition and collaboration in science. Rosalind Franklin's story is now an integral part of that history. Her contribution, once obscured, is now celebrated worldwide.